All right, it's showed up. Okay. So, good morning. Thank you all for giving me the opportunity to tell you about the work going on in my lab. Should we turn the lights a little bit in the front just to see those pictures a little bit better? I don't know. Is that a possibility? All right. It's just a little dark. But anyway, so my lab is relatively new. We opened in July 2019, actually at the University of Alabama, Birmingham. And we moved last week, actually, to UMass Gen Medical School, mostly for family reasons, to be closer to our family in the Northeast. And yeah, so forgive me if I say anything confusing because I rode in a car all week last week with 3 kids under 5. But we're here now, and we moved into our apartment. And so my lab has only been working on Down syndrome for about a year now, but we've been working on neurodevelopmental disorders since well before my lab started when I was a postdoc. And so we work on zebrafish. And these are the same zebrafish that you see in a pet store. I'm gonna hide this thing. It's fine. Hopefully it doesn't disrupt anybody. I saw that you were able to do that. But the animals that I work on are very, very small. So you can see them here at the age we study, which is just about a week old, in a 96-well plate. So there's almost 100 animals in these standard plate sizes, and you can see that compared to a penny here. And the reason we do that is we can get large amounts of data. We can study many genes, we can study drug molecules. People do drug screens in these animals in plate formats, for example. And zebrafish share over 70% of the genes with humans, even though they're fish, they're very small. And you don't think of fish as being that similar, but the genes have quite a bit of similarity, and the proteins they encode tend to be conserved. So one could actually develop a drug for a zebrafish protein that impacted and bound to a zebrafish protein, and it could have relevance to a human. That's not to say it's true in every case, but in a lot of cases. I think I saw that 85% of the disease-associated proteins are actually found in fish. So the fish, as you know, it is a couple months of age. We look at the larval stage of this. As zebrafish are great for developmental studies, and Down syndrome is a developmental disorder that impacts all systems in the body. And these are great because they develop outside the mother, unlike mammalian species. So they're fertilized, and then you can watch every process in development occurring. So in about 36 hours, they have a beating heart. They have a lot of organs that you can imagine, and you can look to see the brain develop over this time period and onward. And we study animals around a week of age mostly, but we are also interested in those around 3 weeks to 4 weeks of age, because their brain continues to develop and they gain new behaviors and new capabilities. When they're quite young, they don't have as many capabilities, but they still have many cell types, and you can study the development of the brain. So I'm optimistic that we can use zebrafish to try to be able to develop drug treatments, and that's a main goal for my lab. This is sort of a hype piece, but it is based on a Nature Medicine article from from 2019. And people really like zebrafish in the rare disease community because they have these random rare mutations that they're trying to study, and you can make a zebrafish with the mutation that your child has. And in this case, they actually tested drug treatments and saved the kid. And this was at Children's Hospital of Philadelphia. So I think that there's an opportunity to be able to use zebrafish, even though they aren't human cells or mammalian. And there's quite a few drugs in clinical trials for neurodevelopmental disorders. There's just one for an epilepsy, Dravet syndrome, but I think that's going to change as we go forward. The brain is obviously quite a bit smaller, but that's also true of mouse models, that it is quite a bit smaller than the human brain, but they're vertebrates. So a lot of the structures form in similar ways, and there's sort of conservation of these developmental processes. The green area is where the cortex hippocampus is, and that's certainly a lot smaller in both fish and mice than in humans, but there are cell types that are similar and a lot of shared mechanisms. So we look a little bit closer at the zebrafish brain. We have a cerebellum, a hindbrain, sort of motor output areas. The tectum region is visual, and the forebrain here at the front is where there's the hippocampus and cortex-like cell types and areas. And if we just look around at there's a thalamus and hypothalamus too as well, in regions you may have heard of. They're just a lot bigger in the fish compared to the forebrain. In humans, the forebrain is just outgrown all of these other structures. The other amazing thing about zebrafish is they're completely transparent at these ages. So this is not data from my lab here, but it is a method that we have accessible if we were interested in it, which is this calcium imaging. So you can see all the neurons firing. So you can give a sensory stimulation to the animal and just basically not watch it think, but you can watch all of the occurrences of the neurons firing throughout the whole brain, which is really exciting for being able to understand the brain. In my lab, we are really interested in brain activity, but we err on the side of a method that's a bit more high throughput because we're interested in looking at drugs and many, many gene changes. And the calcium imaging method is fairly slow and labor intensive. And so the method that we use, we get these basically maps. So this is two views. This is like a top view and a side view of a map of the brain. The outline is the brain. So we produce a lot of these map. You could either be comparing a genetic change or a drug treatment to controls in this map. And what we see here is magenta would be lower activity in that brain area. Green would be higher activity in comparison to the controls. So a little bit about how that method works. Basically, how it works is calcium flows into the neuron. And then as that calcium flows in, this tag gets put on this protein called ERK. So it's a phosphate group. It basically gets tagged in response to calcium. So about 10 seconds after the calcium goes in, this little tag goes on to this protein ERK. And then we have ways to be able to detect the tagged and the untagged form. And so you can see basically what neurons were active. And it's sort of a summary of the last 10 minutes of that brain function of the fish. So we have our fish swimming all around, and we kill them very quickly so that we can just basically capture the brain state of the animals. And then we can image about a couple hundred of these brains every night. So at least it only takes a couple minutes per brain. So you get a whole 3D image of the whole brain. And then we computationally put these all into the same 3D space so you can compare them. So you basically rotate and turn these brains until they're all comparable. And from there, you can look at differences between the groups. So as I said, you can get the activity increased and activity decreased areas when you do that. And you can also look at structural changes. So here is talking about, changes in proliferation. And you can find out if there are areas that were larger or smaller, because as the brain has to rotate and shift to fit into this reference framework, you can find out if it had to stretch or shrink to fit into this more like normal framework. So we use this is one of the main methods we use in our lab every day. We also do zebrafish behavior, and we build our own behavioral setups. We have 12 of these in our lab, and we can modify them as we need. And when we do behavior, we're not thinking like, oh, we have an autistic zebrafish and we're trying to see that it's behaving like that. And that's not what we're looking to see if, how the brain is impacted or the behavior is impacted as a sort of a proxy. Does this gene or drug impact the brain, or does it even impact the muscle? For example, you could detect muscular changes as well. So in these systems, it's about a 4-foot tall box made of plastic, and we have a sound stimulation and a light stimulation, so we can look at sensory changes. And there's an infrared light that animals can't see, so we can monitor them through the night and look to see if the changes in sleep, so that fish have circadian rhythms and a form of sleep. So you can't do anything like learning or memory in this type of plate format, but you can do sort of large scale tests to look at the behavioral fingerprint that is occurring. And fish at this age also do have some other behaviors. They do have hunting. They have some things that are more complex, but not in the small wells of a 96 well plate. And as I mentioned, when they get a little older, they gain the learning and memory capabilities and social behavior. And we're really interested in the next few months that we move the lab up to set up the learning and memory assays in fish, which are well established in the field, but we want to set that up as a way to test our Down syndrome models. And we already have social interaction. So this is just a fun video, so you can see what the fish look like when they're interacting. So these are 3 weeks old, so they're still quite small. You can fit 20 of them in one box, so you can do 20 times 12 just in the afternoon. And these are fish that can't see each other. So there's a divider that's white, and they can't see the stimulus. And then if we remove that stimulus, you can see they really want to spend time with each other. They're just going crazy on the sides over near the stimulus fish. So that is sort of a form of social interaction. If you go snorkeling, you see that fish do like interact with each other. And we are interested in how these behaviors come online, like what is different between 2 weeks and 4 weeks when they now can remember and learn what changed in the brain. So that's one thing that we're looking at in our lab. But I'm really interested in drug development and how we can use zebrafish to discover treatments. We don't have any yet, but that's sort of where we're going with our lab as the big picture. And I think there's 2 pieces to this. We need to understand our models. So we need the characteristics, the phenotypes. So we need to understand what is the aspects of the model that we're looking at. And we also are working on new methods for drug discovery. So on the side of modeling these various disorders, over the past few years, I was mostly working on autism and schizophrenia genetic mutations, but then I switched over to doing Down syndrome work, and we're finishing up those other projects over about a year ago. So we use this brain activity mapping and behavioral methods, and then here I mentioned transcriptomics. That's a way to get a more complete understanding of the molecular changes that happened in response to the drug or the gene. So you can look at the levels of RNA and how those change to get a sense of what's going on that genetic level, rather than just a picture of the brain map. And we also are working on computational drug discovery methods. My PhD work was actually in protein modeling before I went into zebrafish neurodevelopment, and we're bringing those 2 things together in my lab. So we're working on new ways to be able to model and predict drug molecules. And the idea is, big picture, is that we can get targets from our studies of the genetic models and then feed those targets into drug discovery. And then zebrafish is relatively high throughput, so we can test quite a number of these predicted molecules and see if any of them can change a phenotype. Using our methods, we can look to see if maybe it erases a phenotype that we see in brain structure or brain activity. So sort of a really high resolution method to see if we actually had an impact that we thought we did. So over the last few years, I've been working on autism and schizophrenia. This is just to give you a sense of the numbers we can look at. So these are the numbers of zebrafish lines that we have studied and we're basically done with in just a period of 4 years. And prior to that, we looked at over 150 genes linked to schizophrenia when I was a postdoc. So you can do a lot. That's the whole point. And this is relevant because in Down syndrome, there's over 150 genes on chromosome 21. So we don't know which genes actually impact brain development. So our goals for Down syndrome studies, the big one is which genes actually impact brain development. And Bill Mobley talked about ASOs for APP yesterday. If one actually knew which genes were really critical to some of the neurodevelopmental features we observe, one could actually potentially target that with an ASO rather than small molecule like we were talking about. Which of these genes impact learning and memory, not just brain development? And then what are the mechanisms? And are any of these druggable mechanisms? There are big advantages to small molecule drugs if one can get them. So it's worth trying to see if we can find mechanisms that are particularly druggable. Some proteins on chromosome 21 are, but some probably are not. And then can we use this computational drug discovery? So first, how are we building models of Down syndrome in fish? It's actually a bit of a challenge because the genes that are on chromosome 21 in humans are spread all across the different chromosomes in fish. In mice, there's actually 3 chunks of them. And so people have been able to, in mice, that's a more limited number, so you can make an extra copy. So in fish, we can't do this. It's just not reasonable because they're all spread around, even though the genes are there and they have high conservation. So we're looking at the genes individually, and we're overexpressing we're increasing the levels. And so we're using the system, you don't need to worry about the name, but it's called GAL4 UAS. And the idea is that we can control the expression of the gene in various different cell types. So here he mentioned neurons and different other types of cells in the brain. We can control it in that way or in subtypes of neurons. Yeah, so basically the in orange there is a control element that says we want to turn on expression of GAL4 in this type of neuron. So right here, I'm showing you just pretty brains that GFP, the green fluorescent protein, has been turned on in various subtypes of neurons across a fish brain. So one can actually control the expression in specific regions. And then this produces the protein GAL4, and then it binds to another control element, this UAS, and then that controls your gene of interest. And so this is the way we're overexpressing these genes. And one thing that's really important here is we don't want to make it like a 10-fold overexpression. In humans, it's just one extra copy, so it's like 1.5-fold. So over the past year, as we were setting up our system, we optimized that so we're not getting a really high level. We're getting, around 2-fold overexpression of the genes of interest. Here is our fish with the system in place expressing a red fluorescent protein across the whole brain. So you see the red in the brain there, and we've checked the levels, and it's not extremely high. It's not well over what one would expect. So we're interested in looking at the genes on chromosome 21, as I mentioned, but which genes and which cell types. So we want to look at the whole body. We want to look at dividing neurons. We want to look at later neurons, because they're actually done dividing by the time the child is born too. So it would be nice to see if there are phenotypes that show up later. We could be able to target potentially and specific neuron subtypes. But now, what genes? So when I first started thinking about Down syndrome, I was trying to look through all the old literature, and there are these cases of partial trisomy that you could use to sort of pinpoint what areas might be more important. There's this thing called the Down syndrome critical region. That is certainly not the only area that's important for Down syndrome. And there's actually another area where people with recognizable Down syndrome have a triplication according to this old literature, and that's up in that area B. And there's also people with Alzheimer's that have no Down syndrome and no intellectual disability. So this area around APP that Bill Moley was talking about, this APP triplication, these people don't have Down syndrome. So the genes in there doesn't mean they don't impact Down syndrome and make it worse or change some of the features of it, but they're not the like driving force that is causing all these developmental changes that we see. So we're really interested in these like critical genes that are most important to these features. So there's craniofacial features, the brain development. So I decided to initially focus on region C and E here. And one interesting thing about region C is that all the most commonly used mouse models don't even include these genes, so the majority of research in the last 30 years hasn't even included research on those genes because they didn't triplicate that part. And so we picked about 20 genes, and we've made the zebrafish lines that have them under control of this in this GAL4 UAS system. So we've produced these lines, and we're about to start our studies where we're looking to see the impacts on the brain development for each of these genes individually. We can also do combinations of them potentially in the future. We have one gene we're really interested in, and this is in this Down syndrome critical area. And this work is being led by a postdoctoral fellow in my lab, Anna Moyer. She was with Roger Reeves at Johns Hopkins, who was one of the first people to make a Down syndrome mouse model many, many years ago. And her PhD work, so she graduated and then she came to my lab about a year ago to start these projects in fish. She was looking for genes, she looked at 160 genes all across chromosome 21, which ones impact this signaling pathway called sonic hedgehog. I'm not going to talk about what sonic hedgehog is, but basically it's a developmental signaling pathway that disruptions to it is consistent with many of the features we see with heart defects and the craniofacial changes and the brain developmental changes. It's probably the only thing, but it seems like an important signaling pathway that might be disrupted. So Anna was looking for genes that had an impact on that. So she did a bunch of assays. So you don't need to worry about the data or read the assays, but she did 4 different assays and found only a very tiny handful of genes that actually impacted it across all 4 assays that really looked like this is probably a gene that actually impacts this mechanism. And one of them is this gene HMGN1 that we're really interested in. And here I mentioned chromatin packing, and what HMGN1 does actually is it promotes an open state of the chromatin. So this is a very tiny protein, and it affects the chromatin packing. And that's going to cause effects, like he said, across the entire genome. So you're going to see changes, all sorts of different changes to other genes that aren't on chromosome 21. And we're interested in what those are, and if any of those mechanisms that are disrupted might be druggable. So we have preliminary data starting to look at what HMGN1 does in fish. We're also looking at all those other genes I mentioned, but this is the one we started with because we were really excited about this particular one. And what we found is that when you overexpress HMGN1, it does impact brain development. So we have 2 different experiments there. A and B are just basically different ways we introduced the overexpression of HMGN1. One was that system that does the slight increase, that's in the first panel in A. And then another, Anna injected the single cell. So fish are great because you can just put anything you want into that very first cell, and then it impacts the as it's dividing. So you can really make developmental perturbations in that way. So, but the take home is that the brain structure is disrupted in both of those cases compared to controls. And it seems to be a smaller brain, and it does seem to be potentially in areas that are compatible with what we see in humans, and the behavior is also disrupted. Not that that really means anything, because again, we're not modeling Down syndrome, we're just trying to understand the gene, but it does have an impact on the brain. And so we've done some sequencing studies to try to see what molecular changes are occurring. And this is all very new, but it does seem that there is a big impact on mitochondria. And that is actually something that people in the field have talked about before, but not in connection to this HMGN1 gene. So people think that mitochondria and oxidative phosphorylation is something that's really important in Down syndrome, but it wasn't in relation to this gene. There are a number of other genes on the chromosome that are involved with the numbers of mitochondria and the functions of them. And so right now I'm leaning towards this being an exciting and worthwhile hypothesis for trying to figure that out. And I didn't personally expect HMGN1 to have that impact. It has impact on some other interesting genes, and now we're trying to figure out whether this particular finding is actually what causes the brain structural phenotypes, or if it's sort of a downstream outcome of the brain structural phenotypes. What is the initial change downstream of HMGN1 that causes this brain structure to occur? So actually, it seems like HMGN1 is changing the levels of another chromosome 21 gene that's involved in mitochondrial biogenesis. So this might also be important to consider synergy of genes on the chromosome, and do they work together? And maybe, if you have 2 hits, right, you have HMGN1 and this NRIP, then it's probably even stronger impacts on the mitochondria. So obviously we're going to follow that up in the future and try to figure out, more about that. The big picture, we can study many genes and actually many drug molecules, even though we haven't gotten too far on that in the Down syndrome area, since we've only been working on it for a year now. Using fish because they're pretty high throughput. This HMGN1 gene is really of great interest to us for its role in developmental signaling and brain development. And we have these new computational drug discovery methods. I didn't talk about the methods themselves, but that's sort of where we're going. We're interested in using these models to develop drugs, particularly computational methods. And we can study learning and memory in fish at 4 weeks, and we're going to set up those assays and test our models in those as well. And sort of the long-term, sort of big picture, I'm interested in what cell types are involved in learning and memory that are impacted in Down syndrome models, in our fish models. And is it possible to find molecules that can increase the activity of those cells or the synaptic connectivity of those cells? Is there a way to sort of improve that aspect? So we have lots of funding, but in particular, we have funding from the Jerome Lejeune Foundation for supporting Anna, my postdoc. So this is funding for the whole lab for a bunch of different stuff, but the Lejeune Foundation specifically supported her Down syndrome work. And thank the lab. It's going to change as we move to UMass, but a few of the people are going along with me, including Anna working on the Down syndrome work. Thank you.